of the liver. The liver also functions in removing old red blood cells. We talked about this when we looked at the cardiovascular system. It's going to break down hemoglobin, liberate that bilirubin, which is a byproduct, and convert that into bile salts or bile pigments, which can partially be eliminated from the body, but also recycled, which we spoke about, to create new hemoglobin as well. The liver also is important in synthesizing plasma proteins. So think about your clotting factors that we talked about that are always circulating in the blood in their inactivated form. They are made in the liver. Think about hormones that are circulating in the blood in their inactivated form, like angiotensinogen. That is also made in the liver. A whole host of plasma proteins are made in the liver, circulating in the blood. Albumin is another important one that's responsible for our colloid osmotic pressure in the plasma as well. And then finally, the secretion and modification of hormones is also brought about by the liver. Okay. This brings us to speaking about the biliary system. We talked a lot about the actual flow of bile, where it's made in the liver, where it's stored in the gallbladder. So we know that the liver makes bile, specifically the acidic cells in the liver, that is converted into, communicated rather into the common hepatic duct, which is then sent into the cystic duct and stored into the gallbladder. And then that is released from the gallbladder into the common bile duct, which empties into the duodenum depending on certain stimuli that are met depending on the makeup of that meal. So another way to say that is if we had a really large fatty meal, the gallbladder would be stimulated to release more bile into the duodenum. If we had a meal that was not as rich in fat, then the gallbladder would not stimulate as much bile to be released into the duodenum. So depending on the makeup of our meal, bile is released into the duodenum to break down that fat. So the role of bile is really the absorption, the breaking down of fat so they can be absorbed. And so those two openings are communicated together. So it joins with the pancreatic duct. So both the common bile duct and the pancreatic duct join together to form the ampulla of Vater. So the ampulla of Vater is the opening into the duodenum, and that is regulated or moderated by the sphincter of Oddi. So the sphincter of Oddi is a smooth muscle band that wraps around the ampulla of Vater and regulates how often this secretion is released into the duodenum. Lastly here, we'll look at the internal anatomy of the liver. So the materials that are used to make bile are taken up from the blood via these sinusoids. So there are certain sinusoids that are in communication with the vessels in the liver. And the hepatocytes, which are the cells of the liver, take those components, they synthesize this bile, and then that bile is released into these canaliculi. So canaliculi is a term that we've heard before when we talked about the structure of the eye and the lacrimal apparatus of the eye. And a canaliculi is basically nothing but a duct. So it's a small duct that takes that bile and opens it on the opposite side of the sinusoids, okay? And then they can drain that bile into the right and left hepatic ducts, which eventually communicate to the common hepatic duct and eventually to the common bile duct. Okay, and the last thing here on this slide is a really nice overview. So if you took nothing away from this entire packet, I would go back to this slide and generate a really concise study table from this slide. So this is just pointing out each of the organs of the GI system and then the overall function of those organs. So a really nice place to start when creating a study tool. You can certainly add some other details that we mentioned here and kind of focus in on this as a summary study tool. All right, so that is the end of that first packet. Let's see, we'll go ahead and move on to the second packet. I'll see if there are any questions. I don't see anything just yet. Okay, so looking at absorption, we're going to look at each of the macromolecules that we have spoken about, carbohydrates, proteins, and lipids, and we're going to explain the exact mechanism of how these are absorbed. Starting off with carbohydrates. So these are all of the nutrients and molecules that we ingest through our diet. We've got carbohydrates. Those are digested or broken down into their smaller parts, which we know to be glucose, right? That's the monosaccharide that makes up carbohydrates. Proteins are broken down into amino acids. Lipids are broken down into triglycerides, and then further broken down into fatty acids. Vitamins such as A, D, E, K, vitamin B, and so on, those are pretty much absorbed in their natural state, so are minerals. And then water, we know, is also absorbed readily across the GI tract. So this is going to call us to remember a few of the transport mechanisms that we talked about way back in AMP1. So the first one is active transport. So we know active transport requires ATP. One of the important mechanisms of active transport is the sodium potassium, the sodium potassium pump, or the sodium potassium ATP pump, which is pumping sodium out of the cell and potassium into the cell. So 3 sodium out, 2 potassium in, and this is usually found on the basolateral membrane of the cell. We looked at the various examples of how the sodium potassium pump on the basolateral surface brings about many different versions of absorption. So absorption in parts of the kidneys, the absorption in parts of the liver that we'll talk about. So the existence of the sodium potassium pump on the basolateral membrane is going to be really important to absorbing a lot of other nutrients and substances on the apical membrane. So because sodium is being pumped out of the cell, this creates an environment inside the cell where there's very little sodium. And so that is a gradient for sodium to move into the apical surface of the cell passively, right? We've got a little bit of sodium inside the cell because it's being pumped out the basolateral surface. So we have a gradient now where there's more sodium outside the apical surface, less sodium in the cell itself. And so sodium can passively move into the cell. And then the energy that is liberated from that process can be used to bring other substances into the cell as well. We describe this as being secondary active transport. So it's going to call us to remember some of these other mechanisms of transport that we talked about before.